micromolecules, and lipids are insoluble in water. So they're water insoluble biomolecules, but they're soluble in organic solvents. Okay. So lipids are generally the esters of long chain fatty acids and alcohol. Okay. So lipids, as we discussed in the cell membrane, so lipids plays very important role in the organization of cell membranes, right? And lipids are the important molecules for signal transduction, and fuel molecules means lipids are the energy storage molecules. Okay. And some lipids responsible for hormone synthesis, and some lipids are intracellular messengers. Okay. So lipids generally made up of carbon, hydrogen, oxygen elements, but the number of oxygen elements are very limited. Okay. So lipids are the constants of cell membrane. Lipids regulates membrane's permeability. You know, lipids allow only certain molecules, and lipids diffuse only hydrophobic molecules. Okay, and lipids protects internal organs. Okay, and it insulates the organisms. It keeps the body warm. It helps to maintain temperature of the body. Okay, and lipids are the source of fat soluble vitamins. Fat soluble vitamins are vitamin A, vitamin D, vitamin E, and vitamin K. These are fat soluble vitamins. Okay. So next, some essential fatty acids are useful for the transport of cholesterol, formation of lipoproteins, etc. And phospholipids in mitochondria are responsible for transport of electron transport chain components. In the metabolism chapter, we'll discuss that. Okay. So phospholipids are very important in cell membrane as well as mitochondrial membrane. Next, accumulation of fat in the liver prevented by phospholipids. Nothing but phospholipids are the good lipids. These lipids help to prevent accumulation of excess fat, especially in the liver cells. Okay. And it helps to remove unwanted cholesterol from the body. Okay, mark that. Next, in liver, cholesterol is important for synthesis of bile acids, synthesis of some hormones, synthesis of vitamin D, etc. Okay, cholesterol is the important sterol of higher animals. Okay, especially in the mammals. So these are the specific functions of lipids. So lipid responsible for cell membrane structure, and some lipids are the cofactors for enzyme. Some lipids are signaling molecules. Some lipids involved in pigment synthesis. Some lipids are antioxidant. Example, vitamin E. So vitamin E is antioxidant property. So this vitamin is derived from the lipids. Okay. Remember that. Next, second part, classification of lipids. So based on the biological functions, lipids are classified into two types: storage lipids and structural lipids. Okay. Some lipids are responsible only for storage of energy. Lipids are stored only for energy purpose. Okay. Those are called storage lipids. And some lipids are the major structural element of cell membrane, like phospholipids, sphingolipid, cholesterol. All these are comes under structural lipid. Next, some lipid-derived molecules like signaling molecules, cofactors, pigment. These are miscellaneous lipids. Okay. Next, so lipids are classified based on the structural composition. So all lipids are classified into four types: simple lipids, compound lipids, derived, and miscellaneous lipids. Simple lipids are the esters of fatty acids and glycerol. That's it. These are natural lipids. Okay. Simple lipids contain only alcohol like glycerol. Glycerol plus fatty acid. Okay. Example, waxes, sterol esters, triacyl glycerol. These are the examples of simple lipids. Understood, everyone? Glycerol attach with fatty acids through ester bond. That's why they given the word esters. Okay. Esterification takes place. Second one, compound lipids. These lipids are esters of fatty acids with alcohol with additional groups are present. Okay. Glycerol plus fatty acid plus additional groups like phosphate or nitrogen bases present. Those are called compound lipids. You know, sphingosine, ceramide, cerebroside, all these are the compound lipids. Okay. Next, derived lipids. So these lipids are obtained on hydrolysis of simple and compound lipids. Simple lipids, compound lipids, both are hydrolyzed to form a new form of lipid. Okay. Simple lipid, compound lipid, both are hydrolyzed. Both are broken to form a new variety of lipid called derived lipid. Example, as we discussed now, cholesterol, fatty acids, okay, monoacyl glycerol, diacyl glycerol, steroid hormones, hydrocarbons, ketone bodies. All these are the derived lipids, not a simple lipid or a compound lipid. Okay, so these are the three important types of lipids. Next, some other miscellaneous lipids are there. Means these compounds contain characteristics of lipids. Example, terpenes, hydrocarbons, carotenoids, squalene, etc. These are lipid characteristic compounds. That's why these are called miscellaneous lipids. Look at this classification scheme. Simple lipids, compound lipids, and derived lipids. Follow this slide one. Simple lipid example, triacyl glycerol. You know our adipose tissue stores triacyl glycerol. And phospholipids, cerebrosides, gangliosides, sphingolipids, all these are the structural components of cell membranes. Okay. Understood, everyone? Storage lipids includes fats and oils. Fats and oils contain triacyl glycerol. Waxes contain esters of long chain saturated and unsaturated fatty acid with alcohol. So triacyl glycerol is the neutral lipid. You know, adipocytes stores triacyl glycerol for energy purpose. Triacyl glycerol is stored only for energy purpose. Okay. Look at this. Adipocyte stores triacylglycerol. Corn seeds also have triacylglycerol. Cortical cells have triacylglycerol. Okay. So this is the structure of triacylglycerol. So 3 carbon glycerol. So glycerol is a 3 carbon molecule attached with 3 fatty acids. One glycerol attached with 3 fatty acids. This is called triacylglycerol. Look at this. This is the structure of glycerol. CH2OH, CHOH, CH2OH. 3 carbon molecule. Okay. So glycerol 3 carbon attached to 3 fatty acids. Attached to 3 fatty acids. That is called triacylglycerol. Can you see? The OH of the glycerol attached with OH of the fatty acid through ester bond. Okay. Here 3 fatty acid attached. This is called triacylglycerol. Got it? If only one fatty acid attached to glycerol, that is called monoacylglycerol. Monoacylglycerol. If 2 fatty acids attached to glycerol, called diacylglycerol. Okay. If 3 fatty acids attached to one glycerol, that is triacylglycerol. Okay. Fats and oils contain triacylglycerol. So let's discuss fatty acids. Fatty acids are long chain of hydrocarbons. Fatty acids made up of carbon, hydrogen, oxygen in the proportion of 76% carbon, 12.6% hydrogen, 11.3% oxygen. Okay. The number of carbons ranging from 4 to 36. Okay. Fatty acids are 2 types, saturated and unsaturated fatty acid. Saturated fatty acid means, look at this, long chain hydrocarbon does not contain double bond. So between the carbon, no double bond present. Okay, those are saturated. Unsaturated fatty acid means one or more double bonds present in the hydrocarbon chain. Those are called unsaturated fatty acid. Understood, everyone? Any doubts?
Doubts.
For this unsaturated fatty acids, special kind of nomenclature followed. Okay. For example, look at this 18:3. 18 means total number of carbons present in the fatty acid. For example, look at this image. So this is one fatty acid. It contains 18 carbons. Okay, write 18 here. Next 3 means number of double bond. Okay. 1, 2, 3. 3 double bond present. In bracket, write delta 9, 12, 15. 9 means these are the position of double bond. In 9th carbon, 1 double bond present. And in 12th carbon, 1 double bond present. And in the 15th carbon, 1 double bond present. Total 3 double bonds present in this fatty acid. Okay. So this is the nomenclature of unsaturated fatty acids. Okay. So 14 carbon fatty acids are tetradecanoic acid. 16 carbon fatty acids are called hexadecanoic acid. So on. Okay. Same nomenclature formula. Here some fatty acids example given. Lauric acid 12 carbon compound. Oleic acid 18 carbon. Okay. Remember only few fatty acid. Mark this. arachidonic acid 20 carbon, linolenic acid 18, linolenic acid, linoleic acid, oleic acid, palmitoleic acid contain 16 carbon. Okay, so oleic acid, linoleic acid, linolentic acid, these are contain 18 carbons. Okay, remember how many carbon present in these fatty acids. That's it. No need to worry about the structural details and all. Remember how many carbons present and whether they are saturated or unsaturated. That you need to remember. Okay. Next, saturated fatty acids form linear structure. Okay. Did you observe here? Structure is linear. Here, one double bond present here. Right. So this is also similar to the linear structure. So this is transposition. Okay. Cisposition forms curved structure. Okay. Cisposition, this is double bond. Okay. Cis double bond form curved structure. Trans double bond form linear structure. This is the difference. Mark this. Cis double bond in the fatty acid forms curved structure. Trans fatty acids have linear structure. Okay, so this is the different kind of positional isomers. Saturated fatty acid gives viscous nature, unsaturated fatty acid gives fluid nature. That's why cell membrane phospholipids contains one saturated, one unsaturated fatty acid type. Okay. Waxes contain long chain of saturated and unsaturated fatty acid with long chain alcohols. Those are called waxes. Waxed esters of long chain saturated, long chain unsaturated fatty acid with long chain alcohol. So waxes protects the skin and hair. It prevents the excess water evaporation in plants. In plants, cuticle present, that is the waxy layer. Okay, and it is the cheap storage fuel for some of the microbes. And waxes is used in industries, pharmaceuticals, and cosmetics. Okay. And lipids present in the cell membrane, we already discussed it. Phospholipids, sphingolipid, and cholesterol, right? Okay. Same information given here, you can follow this. So this is the phospholipid formation. See? So this is the one glycerol. So glycerol attached with one saturated fatty acid, one unsaturated fatty acid, one functional group. For example, phosphate haded. Right? So this is phospholipid. So next, phosphate attached to the Ed group. So it may be H or phosphatidyl ethanolamine or phosphatidylcholine, serine, glycerol, anything. Okay, so that gives the specific kind of lipids, phospholipids. Fine. Same thing given here. What we discussed in the membrane lipids. Fine. Next, galactolipids in chloroplast. Around 70 to 80% of lipids in thylakoid membrane contain galactolipids. Okay. So in sulfolipid, sulfate group is the functional group. It is also present in chloroplast. Okay. Next, archaebacteria have special kind of lipids because archaebacteria live in extreme habitat in high temperature, in high salty area. Archaebacteria are live. Okay, so these bacteria have ether lipids. Ether lipids. Okay, mark this. Ether lipids are also called plasminogen. So these are common in vertebrate heart tissues, also found in some protozoans and anaerobic bacteria. Plasmalogen. This is similar to the structure of phosphatidyl ethanolamine. Original ether lipids found in Archaebacteria. Modified ether lipids found in some protozoans, anaerobic bacteria, and vertebral heart tissues. Okay. No need to worry about this. Sphingolipids, we already discussed that. Ceramide, cerebroside, ganglioside, right? Glycosphingolipids, etc. For example, see, gangliosides as determinants of blood group. For example, see, ceramide, okay, with 5 sugars present here. Okay, ceramide with these 5 sugars form ganglioside, right? This ganglioside present in membrane of the red blood cells. Okay, so this ganglioside is the O antigen. Okay, so if additional N-acetyl galactosamine added, then it becomes A antigen. Okay, if one more sugar like galactose added, then it becomes B antigen. Okay, so if RBC membrane have this ganglioside, that is O antigen. If it contain this ganglioside, is N-acetyl galactosamine containing ganglioside, additional sugar added. So this is the A antigen determinant. This is B antigen. So like that, so different blood groups present in the human. Okay, in humans, different kind of blood groups are present. Even positive blood, negative blood, A blood group, B, AB, and O. Because of these antigens present on the RBC membrane. If RBC membrane have this ganglioside, so that blood group is O blood group. Okay. So if the RBC membrane contain this ganglioside, then it is blood group A. Okay. So if the RBC membrane contain this ganglioside, then it is B blood group. See, just one sugar determines the kind of blood group. Okay. So if the blood group is AB means RBC membrane contain both A and B antigens. Blood group O does not contain A and B antigen. It contains this O antigen. Understood? Just N-acetyl galactosamine and galactose determines the kind of the blood groups, whether A, B, or O blood group. Okay, understood everyone? Next, sterol. So best example of sterol is cholesterol. Cholesterol is important cell membrane component. And cholesterol is important for bile acid synthesis, vitamin D synthesis, hormone synthesis, etc. Okay? And most of the cholesterol synthesized by the liver. Okay. So actually unsaturated fatty acids reduce the level of cholesterol in the blood. Okay. So increase the synthesis of cholesterol leads to heart diseases. Okay. See, high density lipoprotein, low density lipoprotein, very low density lipoproteins are present. Okay, based on the densities of lipoproteins. Okay. So low density lipoproteins transport cholesterol from liver to the blood and blood to the tissues. Okay. So now excess cholesterol deposited in the tissues. So those cholesterols are called bad cholesterols. So LDL transports always bad cholesterol. Okay. But high density lipoprotein transport cholesterol from blood to the liver. In liver, cholesterol is metabolized to form bile acids, steroid hormones. Okay. See, low density lipoprotein increase cholesterol in the blood. That increases the chance of heart attack. High density lipoproteins, okay, decrease cholesterol in the blood, and that lowers the risk of heart attack. Mark this concept. So these are the cholesterol transporters. Any doubts? So this is the structure of the bile acid. The bile acid is synthesized from the cholesterol in liver. Next, essential and non-essential fatty acids. Essential fatty acids means our body cannot synthesize these fatty acids. These are consumed through the diet. Through food, we consume these essential fatty acids. Example, linoleic acid, linolenic acid, and arachidonic acid. Mark this. So these essential fatty acids are important for blood clotting function, inflammations, okay? Because all these are responsible for the synthesis of prostaglandins, leukotrienes, thromboxanes, etc. Okay, so it is responsible for proper growth and development of the body. Okay, next, trans fatty acids. Actually, these are formed by hydrogenation of normal fatty acids like cis fatty acid. Okay, so trans fatty acids increase the level of low density lipoprotein that increases the blood cholesterol level. Okay, trans fatty acids are formed by hydrogenation process. So normal whatever vegetable oils we use, so normal oils contain cis fat. Okay, on continuous boiling of the oil, cis fat converted into trans fat. Cis fat converted into trans fat on continuous boiling. Okay, this trans fat increases the LDL protein. That increases the bad cholesterol. That's why junk foods are unhealthy. Got it? That's it. So this is about the overall classification of the lipids. Okay. Some say derived lipids, additional lipids are given. No need to worry about this. This is the extended concept of lipids. Okay. Some examples given. Go through this. See, vitamin D synthesized from the cholesterol. So it helps to regulate calcium metabolism. Vitamin D is important for calcium metabolism. Okay. See, retinol comes from carotene. It is also lipid derived molecule. So vitamin E, it is an antioxidant. It is also derived from the lipids. Okay. And warfarin, it is a blood anticoagulant. It is also derived from the lipid. Vitamin K, it's a blood clotting factor. It is also derived from the lipids. Okay. Understood everyone? So carotenoids. These are the colorful isoprenes. Isoprenes are the fatty acid derived molecule. So long chain of hydrocarbons present. Also, long chain of hydrocarbons present. These are the fatty acid derived molecules. Carotenoids are the pigments. Okay. See, brighter red color, bright yellow colored pigment. So these pigments are the derivatives of lipids. Understood? Clear, Vandana? Prisha, understood this?
Yes, ma'am.
Okay.
Yes, ma'am.
Here,